Video we will be studying about the two important blood group systems which are most commonly used as far as the blood transfusions and clinical importance is concerned. One is ABO system of blood grouping and second one is RH system of blood grouping. So we will be studying these two types of blood group systems. So first I will start with the ABO system of blood grouping. So first one, which is ABO system of blood grouping. ABO system. So I will start with the first one, that's ABO system of the blood grouping. Okay, so a blood group, it's also called as a blood type. Okay, so classification of blood, it is based on either presence or absence of the antigens. Okay, so classification will be based on either presence or absence of antigens. So it all depends upon the antigens, whether the antigen is present or absent. Okay, so classification will be based on the antigens. Okay, so these antigens, these are inherited antigenic substances which are present on the surface of the red blood cells. So RBCs here are important because both the type of blood groups are related to the red blood cells. Okay, so the classification of these blood, it is based on either presence or absence of inherited antigenic substances which are present on the surface of red blood cells. And these antigens, these can be either proteins. So these antigens can be either proteins, these can be glycoproteins, these can be glycoproteins, these can be glycolipids, these can be proteins, glycoproteins or glycolipids. It depends upon the blood group system. So mainly we will be focusing here on the antigens as well as antibodies also. Okay, so it is the most important blood group system. Okay, or it's present which is related to the blood transfusion or which is helpful in the blood transfusion. Okay, and ABO blood types, these are also present in some other animals. For example, these are present in rodents, apes. For example, these are also present in the chimpanzees. These are present in the chimpanzees, bonobos, gorillas. But here we will be focusing on, specially we will be focusing on the blood group system in the humans. Okay, so determination of the ABO blood group, it depends upon the immunological reaction between the antigen and the antibody. So basically there will be the reaction between the antigen and antibody. So it all depends upon the antigen antibody reaction. Okay, so antigens, these are also called as agglutinogens. Okay, so antigens will be called as agglutinogens because they have the capacity to cause the agglutination of the RBCs. When antigens and antibodies, they react together, they cause agglutination. Okay, so if we will go to the history of the ABO system of blood grouping, this was Carl Landsteiner, who discovered the ABO system of blood grouping, and he discovered this in 1901. Okay, then there were other scientists who worked with the Landsteiner, who discovered type AB, okay, in 1902. So Landsteiner, he was also awarded a Nobel Prize for this in 1930, okay, for physiology or medicine for his work. Okay, so now we will go to the antigenic systems. So if I will go to the antigenic systems, I will draw the diagrams of different RBCs with the different blood groups. For example, I will go with the first one, that is blood group. So there are four types of blood group as per this. One is A, second one is B, third one is AB, and fourth one is O. So these are the four types of blood groups as per the ABO system of blood grouping. So now I will go with the first one. So four types of blood groups are there. So I am here drawing the structure of the RBC. For example, this is for A, this is for B, this is for AB, and this one is for O. Okay, so blood group A, it contains antigen A. So it is having antigen A. Blood group B contains antigen B. Blood group AB contains antigen A as well as antigen B. or I can draw them as flags. So A flag, B flag, A flag, B flag, and this, it does not contain any of the flags, or it does not contain any antigen. So O blood group, it is not having any type of the antigens. Okay, so A contains A antigen, B contains B antigen, AB contains antigen A and antigen B, while as O contains no antigen. It is not having any kind of the antigen, so it is devoid of the antigen. We can say it is devoid of the antigen. And A contains antibodies against B. So A will be containing antibodies against B. B contains antibodies against A. So, or we can say that A is having an army for B, B is having an army for A. So these armies, these are called as antibodies. So these are the antibodies. For B, we can also call as, it contains anti-B antibodies, it contains anti-A antibodies. Okay, while as AB, it's not having any type of the army because it is having both the flags. So it does not need any type of the army. You can understand in this way also. While as this one, this contains the army both for A as well as for B. So blood group A, it contains antigen A and antibody, anti-B antibody, whereas blood group B, it contains antigen B and anti-A antibodies. AB contains antigen A, antigen B without any antibody. O contains antigen without no antigen and antibodies A and B. Now where from these antigens are coming, or where from these antibodies are coming? Okay, so this we will be this I will be explaining here, where from these antigens are coming. So I will go with the first one. First one I am going here is blood group O, for example. I am taking the example of the blood group O. So this is the RBC, for example. Okay, this RBC, it will be having the genes, because RBCs are devoid of the nucleus. So not RBC, erythroblasts, immature RBCs. Those immature RBCs contain the genes. And this gene here I am writing is a gene number 19. So this gene number 19, it expresses for fucose transferase. Fucose transferase. And because of this fucose transferase, it transfers a glycoprotein which is called as fucose on the surface of the RBC. So if you will go to the O blood group, so this is the blood group O. So blood group O does not contain any antigen, but it has a presence of fucose. This I am explaining because there is one more type of blood group which is called as the Bombay blood group. So here the blood group O, in blood group O, the premature RBCs, those are the from the gene number 19, they express fucose transferase, which transfers fucose to the surface, and it is an oligosaccharide present on the surface. So this fucose, it is an oligosaccharide. Now I will go to the blood group AB, or I will go to the blood group A first. Now this blood group A, so this fucose is present in everyone. Okay, so already there will be presence of the fucose. So this is the fucose which is present in the blood group A. So fucose is present here. Now from here, so this was gene number 9. From here, gene number 9 or chromosome number 9. So this chromosome number 9, it encodes for N-acetyl galactosamine. N-acetyl galactosamine, or we can say NAG, N-acetyl galactosamine. So this N-acetyl galactosamine, what it does, it transfers NAG to the surface of the fucose. For example, this here is an NAG. So this is NAG. So NAG plus fucose, this forms antigen A. So antigen A is formed by the combination of the fucose and the N-acetyl galactosamine. So if someone will tell you what antigen A is, actually it is the combination of the NAG and the fucose. Okay, now we will go to the second. This was blood group A. Now we will go to the second one, that is blood group B. So this is blood group B, same chromosome number 9. This chromosome number 9, here this encodes for galactose transferase. Galactose transferase. And already there will be presence of the fucose. So fucose is present in everyone. So here is fucose, and now it transfers galactose to the surface. So for example, this here is a galactose. This is a galactose. So the galactose plus fucose, this forms antigen B. So antigen A is formed by NAG plus fucose. Antigen B is formed by galactose plus fucose. Okay, so this was blood group B. Now we have blood group AB. This blood group AB will contain both. So chromosome number 9 will encode for both A as well as B. So this is a fucose, and on one there will be presence of NAG, and on second there will be presence of galactose. So this is galactose, this is NAG. So it contains both the antigens, antigen A as well as antigen B. That's why it's called as blood group B. One more thing which we have to remember here, this F fucose, this is also called as antigen H sometimes. So you can remember here, this is an antigen H. So H substance in the blood group A, H substance in the blood group A gives rise to antigen A. H substance in the blood group B gives rise to antigen B. H substance in blood group AB gives rise to antigen A and antigen B, while as H substance in the O blood group, it does not give rise to either blood group, either antigen A or antigen B. Now we have one more type of blood group, that's called as the Bombay blood group. Bombay blood group. So what this Bombay blood group is, you can see here, this is a blood group O. So Bombay blood group, if you will go for the blood testing, it will show you a blood group O. But what happens in Bombay blood group, it's not having presence of H antigen. So H antigen is not present in case of the Bombay blood group. So in case of the Bombay blood group, there is absence of H antigen on the surface of the RBCs. Okay, so individuals with the rare phenotype, means here the phenotype will be HH. Okay, so in this case phenotype can be different, but here in case of the Bombay blood group, the phenotype is HH. Okay, so they do not have H antigen, because HH it will not express, so they do not have any H antigen, okay, which is present in the blood group O. So as a result, they can't make A or B antigen. So A or B antigen they can't make, so they don't have presence of any antigen, but they have H antigen. They don't have even the H antigen. H antigen is actually fucose, so they also lack this fucose, so that's not present here. While in case of O, there is presence of the H antigen. Okay, so they can't make A or B antigen on their red blood cells, so they have anti-H in the plasma. Okay, so what they can do, they can donate RBC to any member, donate RBCs to any member of the ABO blood group system, but they can't receive blood from any member of the ABO. So we can also call it as a universal donor. It may be called as universal donor because it donates the RBCs or blood to any member of the ABO blood group system but cannot receive. Okay, so in short, you can remember here that H antigen is absent here, and it was first discovered in Bombay, which is now known as Bombay, by Dr. Y.M. Binde. So it is present in almost 0.0004% of the population. So that is the thing which you have to remember. Actually, it happened because of the accident. One lady, she was having an accident. She was rushed to the hospital. In hospital, they checked her blood group because they needed the blood for the transfusion, because they needed to transfuse the blood in her. So they found that blood group is O. So when they transferred blood to her, and that blood showed reaction. Okay, and then when further studies were carried out, they found that this blood group is not, she was not having the H antigen present on the RBCs. That's why this blood group was named Bombay blood group. So it's it is present in only 0.0004% of the population. Okay, and the important thing here is to remember is that they donate RBCs to the any member of ABO blood group system, but they can't receive blood from the any ABO blood group system. Okay, now from this what we have learned, this fucose and all. So there are antigens on the RBCs, A, B, AB, and O. So these are the blood groups. These are the 4 types of blood groups. So antigen present in the A is A, antigen present in the B is B, antigen present in AB is A and B, and 0 is none. And antibodies, these are antigens. Now antibodies present in the A is anti-B, B is anti-A, AB is none, while as O is having AB. That's why O is a universal donor and AB is a universal acceptor. So that is the case. Okay, universal donors and universal acceptors. Now this can give blood to and this can receive blood from. First I will write give blood to and receive blood from. Now A is having the antigen A and antibody B. So whenever you are discussing the blood transfusion, you have to remember one thing which is most important. There is a Landsteiner rule. So Landsteiner rule, what it states that if antigen is present on patient's RBCs, the corresponding antibody, it will not be present in the patient's plasma under the normal conditions. This thing you have to remember. Okay. So once again I will say it, Landsteiner rule. So if antigen is present on a patient RBC, the corresponding antibody for that, for example, this is a RBC, antigen is A, corresponding antibody will not be present here, it will be present on the B. Okay, so B antigen, for B antigen the antibody will be present in A. So that is called as the Landsteiner's rule. And important thing here, because I am going to explain you the who can receive, who can donate. So you have to remember one thing, while transfusing the blood, antigen of donor is considered and the antibody of recipient is considered. Antibody of recipient is considered. This means that antibody of the donor and antigen of the recipient, they are ignored. Okay, so thus RBC of O has no antigen, so agglutination does not occur with any other blood group. So O blood group can be given to any blood group persons, and the people with this blood group are called as universal donors, which I already told you. So we will go through this one first. Now who can donate and who can receive. So first, for example, we have a blood group A. So blood group A can give blood to A and AB. Why to A and AB? Because AB, C, for the donor, can give blood, that means donor. Okay, so in this case, we have to write for A. So blood group A, okay, A and AB, it can give to A and AB. Why to A? Because A is having the A antigen, so it can give to A. Why to AB? Antigen of donor is considered, and in AB there is no antigen, that's why it can donate to AB also. Why not to O? Because O is having what? O is having antibody AB, so that's why here it can't give blood to the O. Second case is same, B can give blood to B and AB. AB can give blood to only AB, and O can give blood to AB, A, B and O. That's why it is a universal donor. Now receive. During the recipient, what we consider? Antibody. Okay. Now A can receive blood from A and O, because we are considering here the antibodies. B can receive blood from B and O. AB can receive blood from all AB, A, B, O. That's why it's a universal recipient. Whereas O can receive blood only from the O, because we are considering the antibodies. It is having both the antibodies. Okay. So this is how the blood transfusions occur. Who can give blood to and who can. Now what is agglutination reaction? I was telling you about the agglutination reactions. For example, this is the RBC, for example. This is the RBC, and this RBC contains A antigen. I am taking the blood group A. type A blood group, okay. Plus, for example, this RBC reacts with an antibody. For example, this is an antibody and this is anti-B antibody, okay. So this one is anti-B antibody in type A blood. So this is blood group A, and in this blood there is presence of a anti-B antibody. What will happen? Will these two react or not? So there will be no agglutination. Why they will not agglutinate? Because this is not the corresponding antibody of the A. This is a different antibody. So there won't be any type of agglutination. That's why A can donate blood to A and AB. Okay, so A can donate blood to A and AB. So there won't be any type of agglutination. So this is the RBC with antigen A, and here there will be antibody. So there is no agglutination reaction. Now, for example, if we will take type A blood again, this is the type A blood. It is having antigen A present on the surface. Now it reacts with the antibody, and now I am taking the antibody, that's anti-A antibody. Anti-A antibody means this antibody will react with the antigen A. So here there will be agglutination, means they both will combine together. So here there will be agglutination, they both will combine together, okay. This is the RBC, it is having antigen A, and here there is an anti-A antibody. So that is the reason why we are not giving blood to, why this A can't donate blood to B, because in B there is presence of anti-A antibody, okay. So this is blood group A and this is anti-A antibody. So these both will react together and they will agglutinate. This reaction is called as agglutination, okay. So this type of reaction, this is called as agglutination reaction. So this is an agglutination reaction which occurs in our body, and this has to be considered during the transfusions. One more thing which we have to remember here, in case of the mismatched transfusion, the transfusion reactions, they occur between the donor's RBC and recipient's plasma, okay, in mismatched transfusion. So the reactions will occur between donor's RBC and the recipient's plasma. So if the donor's plasma contains the agglutins against the recipient's RBC, agglutination does not occur, like here. So here there won't be any type of agglutination. You can see here there is no agglutination reaction, while in the second case agglutination does not occur, okay, because these antibodies are diluted in the recipient's blood. So here there is no agglutination in the first case, okay. And transfusion reactions, there are some transfusion reactions which occur due to ABO incompatibility. For example, transfusion reactions, these are adverse reactions in the body which occur due to transfusion error, that involves transfusion of incompatible or mismatched blood. Sometimes the mismatched blood is transferred, okay. So the reactions, they may be mild, they can only sometimes cause fever, okay. Sometimes they may cause fever, and sometimes they may cause hives, that is a skin disorder that is characterized by itching, or it may be severe leading to renal failure, sometimes renal failure or shock or death. And this occurs only due to incompatibility, ABO incompatibility. So transfusion reactions, what are those reactions? These are the adverse reactions in the body which occur due to transfusion error. For example, this second case, this is an agglutination reaction, okay. So here there is a transfusion error. For example, we are giving the blood of A and there is an anti-A antibody, so there will be agglutination reaction. So what happens in transfusion reactions? These are adverse reactions in the body which occur due to transfusion error that involves transfusion of the mismatched blood. So here there is the transfusion of the mismatched blood, okay. So it may cause fever or hives, or it may be severe, it can cause renal failure, it can cause shock, or it can cause death, okay. That's the thing. Now we will move on to the second part. So this was the ABO system of blood grouping. Now we will move to the second part, which is RH system, or RH system, or which is also called as rhesus system. because of the rhesus antigen. Okay, or it's also called as the D antigen. So this was first found in the rhesus monkey. Okay, so RH system of blood grouping, it has its own importance. What was done to understand this RH system? For example, so the RBCs, red blood cells from the monkey, were taken and they were transfused in. For example, here this is a rabbit. Don't go to the diagrams. Okay, so for example, this is a rabbit. So RBCs from the monkey, they were transferred to the rabbits. I will draw one other diagram. And this is a human. Okay, so RBCs from the monkeys, they were transferred to the rabbit, and what the scientists discovered, that this rabbit developed antibodies against the RBCs from the monkey. Okay, so RBCs from the this monkey, these RBCs from the monkey, they were transferred to the rabbit, and what they noticed that after some period of time, this rabbit developed the antibodies against the RBCs of the monkey. Now these antibodies which the rabbit developed, because it developed the antibodies, monkey RBC antibodies. So now these antibodies, these were transferred to the humans, and what they saw in the humans, that these were transferred to the humans. Okay, so 85% of the population showed reaction, while as the 15% of the population didn't show reaction. And what the scientists then concluded, that these 85% of the human population is Rh positive, that means they contain the Rh antigen, that's why they reacted with these antibodies. While as this 15% of the population, this 15% of the population is Rh negative, that's why they didn't show any type of the reaction, because the antibodies didn't react with anything, because they are Rh negative, that means they are having the absence of the antigen. Okay, Rh positive means they are having the antigen, that's why they reacted with the antibodies which were formed in the rabbit. Okay, and the 15% of the population, they didn't react because they are not having the antigen present in them. Okay, so this experiment was also conducted by the Carl Landsteiner. Okay, now if we will go to the RH system of the blood group in detail. So if we will go to the RH system of blood grouping in detail, I will take one example. So for example, this is an RBC from Rh positive blood of a person, Rh positive. So this is the RBC of the Rh positive person. What do we find in this RBC? There is a presence of a protein. This is a protein which is present in the membrane. We call it as 12-pass membrane protein. 12-pass membrane protein, because it passes through the membrane 12 times. One will be amino group, one will be carboxyl group. Okay, so on this 12-pass membrane protein, there is a presence of an antigen. There is a presence of an antigen, and this antigen is called as antigen D. So Rh positive persons generally have, so there are different types of antigens, almost 35 types of antigens, but 5 among them are most common. One is D, capital C or small c, capital E or small e. So these are the most common or most popular Rh positive antigens, but among all of them, D is most immunogenic and most prevalent. So it is most immunogenic and most prevalent antigen which is present in the blood of the Rh positive persons. Okay, so again on the second loop, there will be presence of the antigen C, capital small c or capital C, and on the fourth, there will be presence of the antigen E or small e. Okay, but most immunogenic, I have shown it like this, it's most immunogenic. So D is most immunogenic. Okay, so Rh positive persons, we can call them as D positive persons also, because actually the Rh positive persons contain the antigen which is called as the D antigen. So they have the presence of D antigen in their blood. So Rh positive, Rh negative persons, they will not have D antigen on their blood. Okay, so the Rh blood group system, it's one of 35 current human blood group systems almost, and it is the most important blood group system after ABO. And Rh blood group system, it consists of 50 defined blood group antigens. Okay, among them there are almost, I told you, there are 5 antigens which are most common: D, C, small c, E and small e. Okay, each of which is called as an Rh factor. We can call them as Rh factors also, and they are designated as D, C, small c, E, small e. Okay, so the type D antigen, it's widely prevalent in the population and it's more antigenic. Okay, I have shown it here. It's more antigenic than other Rh antigens, and anyone who has this type of antigen, it is said to be Rh positive. So a person with a D antigen is called as Rh positive, and a person who is not having D antigen is called as Rh negative person. And this antigen, this was discovered also by the Karl Landsteiner, who discovered the ABO system of blood grouping, and second person who was with him was Alexander Wiener, and they discovered this in 1940, and this was first discovered in a monkey, rhesus macaque, and hence it's also known as Rh factor or rhesus factor. Okay, now if we will go for who can donate blood to and who can receive blood to, and what are the different types of blood groups or antigens which are present. So from Rh positive system of blood grouping, there will be different types of RBCs. So I will name these RBCs. 2, why I have made 8? Because for example, this is A positive, A negative, B positive, B negative, AB positive, AB negative, O positive and O negative. Now this A will be having an antigen A and antigen D, because this is A positive, so it will be having D also. A negative will be having only antigen A. B positive will be having antigen B, antigen D, while as B negative will be having only antigen B, because it is negative, it does not contain antigen D. AB positive, it is having antigen A, antigen B, antigen D, while as AB negative, it is having antigen A, antigen B. O positive, O negative, O positive, it will be having an antigen D, only one antigen will be present, which is antigen D, and O negative, it will not be having any type of the antigen present in the blood, present in the RBCs. Okay, so this is about the Rh. Now we will be studying here who can give and who can receive blood from this. So I will make one table. So first, blood type. Second one is can donate blood to, and third one is can receive blood from, So we have different types here: A positive, O positive, B positive, AB positive. So first I will go to the positives. So it can donate, A positive can donate blood to A positive and B positive. Keep remember those antigens, because when we are donating blood, we consider the antigens. When we receive the blood, we can consider the antibodies. So O positive can donate blood to O positive, A positive, B positive and AB positive. So here it is A positive and AB positive. B positive can donate blood to B positive and AB positive. AB positive, it can donate blood to AB positive. Now we have other 4. Then there is A negative, O negative, B negative and AB negative. Okay, so A negative, it can donate blood to, A negative can donate blood to, A positive, that is the first one, it can donate blood to A negative, O positive, O negative. sorry, actually we were writing about the receiving. So A negative, it can donate blood to A positive, A negative, AB positive, then AB negative. O negative, it can donate blood to all, that's why it is a universal donor. Okay. B negative, it can donate blood to B positive, B negative, AB positive, AB negative, and AB negative, it can donate blood to AB positive and AB negative. Now, second thing is can receive blood from. A positive can receive blood from A positive, A negative, O positive, O negative, because I have already explained you how this is determined. It is based on the donation is based on the presence or absence of the antigens, while receiving the blood we consider the antibodies. So I already explained you about the antigens and antibodies. You can see the previous previous information I have given on them. So second one is O positive can receive blood from O positive and O negative. O positive, O negative. B positive can receive blood from B positive, B negative, O positive, O negative. Okay. AB positive can receive blood from all, that's why it is a universal recipient. AB positive universal recipient, O negative universal donor. A negative and O negative. O negative can receive blood from O negative. B negative can receive blood from B negative, O negative, and it can receive blood from AB negative, A negative, B negative, and O negative. So this is all about the transfusions, how the transfusions occur because of the RH system of blood grouping, because we have to consider it. Almost 85% of the population are RH positive and 15% of the population are RH negative. That's why we are considering this thing here. Now there is one thing which is associated with this, that is erythroblastosis fetalis. That is an important thing. Erythroblastosis fetalis, or it's also called as hemolytic disease of newborn. Hemolytic disease of newborn. It is associated with the RH system of blood grouping, so that's why I will be explaining it here. For example, we have a father and mother. For example, we have RH positive father and RH negative mother, and during pregnancy, for example, a baby, they give rise to a baby and that baby is RH negative. I am taking the example, RH positive baby. Okay, so father is RH positive, mother is RH negative. Okay, so father, he is a positive, mother, she is negative. One of them is positive, one of them is negative. Okay, so what happens during pregnancy? RH negative mother with RH positive baby. So RH positive baby and RH negative mother, because mother is RH negative, so it has formed the RH positive baby. And what happens at delivery? At the time of delivery, what happens? RH positive baby's blood cells, they enter the mother's bloodstream. So these RH positive cells, they can enter into the mother's stream through the placenta, mother's blood through the placenta. So these positive, they will enter into the mother's bloodstream. They can enter at the time of the delivery. Okay, so they will invade RH positive blood cells. So these invading RH positive blood cells, this cause the production of RH antibodies. So what mother will do then? It will produce RH antibodies against this RH positive blood. Okay, so after at the time, there is no problem at the first delivery. After months later, so these RH antibodies, they will remain in the mother's blood. Okay, so if there is another pregnancy at months later, if the baby born is again RH positive, what will happen? RH antibodies which are already present in the mother, so these RH antibodies, they will attack the RH positive baby, the second birth. Okay, so RH antibodies attack the RH positive baby's blood and causing the RH disease. which is called as the hemolytic disease of the newborn. We can call it as hemolytic disease of the newborn. So in short, we can say that what happens in erythroblastosis fetalis, it is a disease of the fetus of it is a disease of fetus and newborn child, which is characterized by the agglutination and phagocytosis of the fetus red blood cells. Okay, and in most of instances of erythroblastosis fetalis, the mother is Rh negative. I have mentioned it here. So in most of the cases, the mother is Rh negative, okay, and father is Rh positive. So Rh negative mother and Rh positive father, okay. So Rh negative mother, Rh negative, Rh positive father, Rh negative mother, okay. In most of the cases, the baby has inherited the Rh positive antigen, okay, from the father, and mother develops anti-Rh antibodies. You can see it here. So if I will explain it again. So in case of erythroblastosis fetalis, what happens that, which is also called as the hemolytic disease of the newborn. So in erythroblastosis fetalis, what happens that, for example, the father is Rh positive and mother is Rh negative, and for example, the baby, it takes the antigen from the father and becomes Rh positive, and this Rh positive baby, the RBCs from the Rh positive baby, they enter into the bloodstream of the mother. Okay, they remain in the bloodstream, and after months, after some time, the antibodies will be developed by the mother, because mother is Rh negative. So antibodies will be developed by the mother. Okay, so antibodies will be developed by mother here. When the antibodies will be developed, they will remain in the mother's bloodstream, and after months after the delivery, after months later, at the second delivery, if the baby is again Rh positive, and because mother is already Rh negative, so this mother has already the presence of Rh antibodies in her blood. So these Rh antibodies, they will attack, where they will attack? They will attack the Rh positive baby. So Rh positive baby will be attacked here. Okay, so Rh positive baby is attacked, and this disease is called as the Rh disease. Sometimes it can kill the mother also, or sometimes it can destroy the RBCs of the newborn, which causes the hemolytic disease of the newborn. Now we can prevent this condition at the first pregnancy. When there is Rh positive baby, when the mother develops antibodies, we can give her the injection here. Okay, we can give her anti-Rh injection, anti-Rh injection. So if we give her anti-Rh injection, these antibodies will be removed from the body, and second delivery will be safe for the mother. Okay, so symptoms which occur in the fetus are generally here. In Rh disease, the symptoms which occur, first is enlarged liver and spleen. Enlarged liver and spleen. Okay, you can see this through the ultrasound. So there will be enlargement of the liver, enlargement of the spleen in the mother. Okay, so symptoms and signs of the newborn. This was for the fetus. So this occurs in the fetus. For the newborn, what happens in the newborn? There will be anemia. Okay, and because of the anemia, there will be pale appearance of the newborn. There can be jaundice or yellow discoloration of the newborn skin, or sclera, or mucous membrane. There will be enlargement of the liver in the newborn. There will be enlargement of the spleen in the newborn. There can be severe edema, build up of fluid. Okay, or there can be dyspnea, or there can be difficulty in the breathing. So these are the symptoms and signs which occur in the newborn. Okay, so this was all about the blood group system. And once again I will explain you, because this is important, this erythroblastosis fetalis, it is most important in this transfusion. This is one of the questions which may be asked in the examinations. So in case of the erythroblastis fetalis, what happens in erythroblastis fetalis, which is also called as the hemolytic disease of the newborn. Okay, so in this, what happens that the mother is Rh positive, mother is Rh negative, and the father is Rh positive. So Rh negative mother, Rh positive father. So at the time of the pregnancy, so they give rise to the Rh positive baby. This Rh positive baby, at the time of the delivery, so through the placenta, these Rh positive antigens, they can cross the placenta and they can go into the mother's bloodstream. Okay, so they can enter into the mother's bloodstream here. Okay, when they enter into the mother's bloodstream, because mother is Rh negative, so the mother will produce antibodies against this antigen. Which antigen? Positive antigen, or D antigen. Okay, so these antibodies will remain inside the mother's bloodstream, and mother months later, when there is a second delivery, and at the time of second delivery, if the child again is, if the fetus again is Rh positive, what will happen? Mother already has Rh antibodies. These antibodies will attack this Rh negative fetus and will cause Rh disease, in which there is enlargement of the liver, enlargement of the spleen. There can be, for the newborn, this for the fetus, in the newborn there can be anemia, there can be jaundice, there can be liver or spleen enlargement, there can be severe edema, there can be dyspnea, or there can be difficulty in the breathing. So this was all about the Rh and ABO system of blood grouping.